we might do is get all the panel of speakers to come up the front, and we're going to have a bit of a Q&A. So what we might do is open it to the floor here while we see if there's any online too. So is anyone would like to ask any questions of Mark?
You spoke about how the donor cells could potentially, or your data certainly suggested, could be very helpful in reducing the incidence of CLAD. Can you just expand on which type of donor cells, and how could we potentially upgrade the right number of donor cells that come across? I presume there's some form of lymphocyte. That's a guess by me.
I think the answer actually links all of the research that you've seen here today. So the specific cell is a lymphocyte, and it's the T cell and the persistence of those donor T cells that seems to be favourable. And what we're going to do next is kind of go back to Lucy's bit of work and see if there's particular combinations of donor and recipient and structural similarity or dissimilarity that predicts whether or not those cells hang around. And it also may be related to the work that Nicole presented, but it may be something about, you know, what these T cells actually look like that they're accepted. We suspect that T cells generally cause harm in the way that Nicole has just described. But there's also this other subset of T cells which are called regulatory T cells, that are good T cells that actually protect against harm. So our next job is basically to drill down, look at the nature of those T cells, and then look at some associations between the donor and the recipient to see if that predicts whether or not they hang around.
How would you get more in there at the time of surgery?
Well, I think part of it is, but if it is related to a certain combination between the donor and the recipient, well, then we try and match towards that at the point of transplant.
And the other question is, can you take some donor bone marrow, so some big chunks of donor cells, and actually have that bone marrow working in the recipient as well? And it's been, again, talked about for a very long time, but there's some work coming out that doing a combination of marrow and lung transplant could help a bit. And we do know that if you have multiple organs, you start just with, you know, a lung, you add in a heart, you have a lung, heart, kidney, or a lung, heart, liver. The mass of tissue seems to actually help you as well. It almost confuses the immune system by just having so much.
I have a question. Thank you so much for all the work that you guys do. It's brilliant. It's creating a future, a hope for the transplant patients and also their families. Greg, I had a question regarding the work you're doing with the AI, with the algorithms, with improving that. You were saying that they're using a number of organs, the heart, the lung, and that. What do you feel like, how much longer do you feel it's going to get to that point to get away from the blue cells under the scope into that? And is it more money? Is it more volunteers participating in that trial? Like, how can we condense that to make that a new gold standard of review?
I think it's essentially reached a point where the science would back it being a good thing to try and do it, and the proper way to do it would be to do it in a randomised trial, and multiple centres, but that would require money to do that. The technology, the concept is real. Lucy will tell us that the science has evolved a bit, and can the science be a bit cleverer to use the very latest technology beyond even the gene chip technology that was used. But it's bringing, as you say, it's taking something that's working and taken years to get there to say, okay, now how do we just explode it? And I think probably a bigger trial that's supported with money would be the way to do that, because they do it all on a shoestring over in Canada.
Can I just add to that? Greg highlighted that this is the work of this nephrologist, Phil Halloran, and I think that there are lots of groups doing similar things to him. But the smart thing that Phil did is that he linked up with a big commercial diagnostic company. So he's basically created the technology and has been able to commercialize it. And it's now been FDA approved in the US, which is the first step for it being routinely clinically used in clinical practice. And that is now being routinely used in renal transplantation. So I think that was the smart bit, is taking the technology but commercializing it and linking with a commercial partner.
But we still need a trial that'll really be what convinces, say, in Australia, the government to ultimately fund it, to say it's not just a good idea and it seems to work. We can actually prove it to work.
There was one question that I noticed online from Bridget Maloney. Malahi, who says, excellent presentations, really informative update. Her question is Atlantis being used within Monash across organisations or more of an open source database? Over to you, Nicole.
So Atlantis has just been in development in the last few months, but we're about to put an MRFF funding application in to try and get it so that it's be at least at 3 different nodes. There'll be a Western Australian node, there'll be a New South Wales node and a Victorian node. At the moment, it will be available if we do get the funding in 4 years' time to be accessed by the public. So that's the big picture.
Can I ask a question? Thank you all for the fantastic work that you do and the great presentations today. So a question for Nicole again, if that's okay. It's a hard question. A really exciting work, and I'm just wondering, have we got any promising targets in relation to developing, designing some therapeutics that can be more specific? I don't know if there's anything you can share on that front.
Yeah, at the human side of things, we're just sort of ramping up that, but we do have some preliminary data in the mouse where we've been able to identify the peptides, the T cells that are recognising those, and we can actually make reagents that can actually pull them out of the circulation, and that's what we're trying to do with the human system. We do have a list of peptides that we're trying to work through. Some of the preliminary data is obviously going to add to that already evaluated list, if you like, but it's certainly something that's going to take 3 to 5 years before we're able to get it to a point where we think we'll be able to use that in a patient system.
I've just been thinking about things, and I just wondered, we've got long-term survivors. Have you compared some of their tissue for any of these?
It's a good question. Is not just to look at the trouble, but to look at what is successful. And certainly, cancer thinking has gone that way a bit to say, what are the inhibitors of rejection? What are the inhibitors of cancer? We haven't specifically done that. I think it's something that is being teased out, I think, with some of the blood work that Steve's been doing, trying to look at the match combinations that do the best and then understand why they do the best in due course. And if we can pin down specific combinations that we should not do and specific combinations we should do, again, perhaps using computer assistance, that will be the way to go. Yeah.
It's just a comment. I remember in the very early days, perhaps even before you joined, we were able to do with the hearts compatibilities. I even recall being told when someone went to sleep, who are we going to choose? Well, you find the one that's got the most compatibilities. And those heart patients said, I will.
So we're certainly...
And also the heart-lung ones seem to.
Yes, so we're certainly aware of we're getting ideas of what are the best matches to have and what are the best ones to do, and we've broken that down a lot from over the years from some of the simple understanding of what was a match or a mismatch. It's just in lungs, you've just got a limited pool of lungs a year, whereas if you want to do a blood transfusion, there's 1,000 units of blood that you could have, and they can go right down to 10 at one end, and you'll only need 4, so you don't even hear about how hard you would a cross match. Whereas if we've only got 3 lungs for you in a year and you're hard to cross match, that could be sometimes quite a challenge for us. But we are pulling that apart, and Steve's work's been very helpful for that.
I was just going to add to that is that, for many decades, we've looked at this HLA molecule as a standalone molecule with many, many amino acids in it. And we're now entering this molecular area where we've realized that it's actually maybe less important the whole molecule, but it's actually the actual sequence of maybe 3, 4, 5, 6 amino acids within that's important. So I think we're getting a better understanding by diving deeper into these HLA molecules at this kind of molecular level.
We've got a few other questions online. So Stephen Wright, who's one of our patients, he has 2 questions. Chimerism, so interesting. Is the amount of donor cells that come with the organ random or controlled? Or do organs get washed prior to transplant? I'm going to answer that, Glenn.
It's random and also controlled. So we try and remove them in as much as that all donor lungs will be flushed through with saline water, for lack of a better word, to try and remove as much of those immune donor cells as possible. But we also know that there are lymph nodes, and Greg talked about marrow. Lymph nodes are full of immune cells, and they're less easy to control, whether they come over with the transplanted lung.
And so Steve also says, back in 2011, I was informed that 50% of recipients are still alive after 5 years. A few years ago, I was told that it improved to 7 years. Is there an update to this stat? Greg.
So we would be expecting, on average, we'd be expecting more than 50% survival at 10 years, more like heading towards 60% 10-year survival if you were transplanted today. So it's not perfect, but it certainly beats having oxygen and running into troubles over 2 months.
A lot of good responses to that question going on in the background there, which is good. So another patient, Karen Pritchard, has asked, do you have an expected timeline re the inhaled tacro, and will there be more trials possibly with those experienced skin cancer issues?
We're just getting started in it. It looks very promising. There's some push to just start someone on it early from transplant, but we've got to be cautious. We've got to be careful and safe. So we're picking a group of people who are particularly troubled by some of the side effects of tacro and don't have an alternative strategy. And we will work with the company to try and roll it out. We're running an Australian study with the other transplant units, so that'll increase the sort of speed at which it gets out. And then I'm sure we'll be looking at a study that will go earlier from transplant, perhaps at 3 months or 6 months after the transplant. But we'll have to see how the results go first.
So just hold on to that microphone, Greg. Jane Fletcher's on the same tack as oral tacro, inhaled tacro. She has asked, does this mode of delivery increase the patient's risk of oral cancers?
So anything that's inhaled, obviously you press the little thing and so you take a breath in and we know from asthma work and that the powder can hit the back of the throat and you have to rinse the mouth out and out it comes. So we'd be thinking there's a bit more of a risk of thrush. There's no known particular toxic effect that I would be thinking of on the tissues. It doesn't cause lung cancer or that when it goes down further. So it's a very good point that we will be always looking for new and different problems, but we'll also be hoping that the technology that sees these tiny little bits of powder flung all the way down to the corners of the lung is the same technology that's been used, in gazillion patients with asthma and that it'll prove safe. But we're certainly going to be testing all that.
Did you want to make a comment about the inhaled steroid story and how we looked at that 20 odd years ago?
Yes, so a long time ago, Helen Whitford was working with us on that one, looking at inhaled steroids, which are used in asthma and that didn't prove to be as successful, although it was harder to work out how much effect you were having because the steroids you can't measure, whereas the tacro you can measure in the blood and in the lung. So that wasn't as good, but there's been a study that did use a nebulizer with cyclosporin, which we don't now use, and that did suggest you had better results in the long term with that. So it's part of an evolution, and again, this is the best product and the best way of delivering it. We've just got to work out whether it does live up to our expectations.
Lucy, you talked about two pieces of important work. One was the threshold levels of HLA and also the incidence of non-HLA antibodies. So it looks like we've got good data. Has any of this had an opportunity yet to get into trials in humans with lung transplantation, or is that planned?
With lung transplantation, the non-HLA not yet. We're just getting started, and I think that Lungitude have agreed to give us some seed funding to support that moving forward. And the
other was the HLA threshold work.
Yeah, so that one absolutely has been rolled out across Australia-wide, and it really dictated what we do on the night of transplant when we're matching donors and recipients, not just in lung transplant recipients, but it changed kidney, pancreas, the way that we do virtual cross-matching across all those different organ groups.
Does it mean that some people who would have otherwise had a positive cross-match and been turned down have now received transplants, and did that go well, or did many of them suddenly develop acute rejection?
Yeah, so virtual cross-match final implementation was in February this year, and so far the data we have, it's very promising. There's been no adverse events that have been reported. And we also, based on Steve's work, developed a plan, if you like, around which patients we actually would perform a flow cytometry cross-match on the night of transplant if they really required it, based on the sort of low level of antibodies that may be borderline. We have only been called on to do that a couple of times, and what we really see is that people do trust the virtual cross-match now, so it has been a very successful rollout, yes.
As clinicians in lung and all of those kidney transplant clinicians, we were pretty terrified that this change, forced change, I might say, for complex reasons, that we weren't able to continue the existing system and it had to change. And there was a lot of discussion about, hey, what if you're wrong? What if if the cutoff's not right? What if we transplant people and get troubles? And they wanted all sorts of backups, and we were part of that as well, saying, well, you have to be able to get us back to the old way if it's not right. But in fact, it's gone swimmingly, and now it's universally sort of, okay, that's just how we do it.
Yeah, and in actual fact, our data is now being used worldwide to sort of roll out virtual cross-match in other countries, and the trust that it actually does work, as long as you have very trained scientists who know their HLA antibodies, because interpreting HLA antibodies is an art form in itself, and it's a very complex process. I think you guys will agree with that.
Well, we might thank our panel for answering questions so well and for their great presentations.